Hello, my students. I'm your biology teacher, Cikgu Govin. Our topic today is leaf structure and function. The subtopic is 2.3, main organ for transpiration. Here are the learning standards. Number 1, justify the necessity of transpiration in plants. Number 2, describe environmental factor that affects the rate of transpiration, like intensity, temperature, air movements, and relative air humidity. Look at these diagrams. These diagrams show the path of water movements in plants. The red arrow indicates the water movements. It starts from the soil here. Root absorb water and mineral salt from the soil. And then at this part, the xylem transport water upwards in the plant stems. And then at the leaf veins transport water to the leaf, and then water evaporates to the atmosphere through stomata. Here is the stomata. Usually stomata is under the lower epidermis. Okay, what is transpiration? Transpiration is a process of water loss in the form of water vapor through evaporation from the plants to the atmosphere. Why is transpiration important in plants? Transpiration in plants helps absorption and transport of water and mineral salt from soils. Transpiration also plays an important role in cooling effects. Water absorbs heat energy from leaf and evaporate as water vapor to give cooling effects to the plants. Transpiration also produces a pulling force that moves the water and mineral salt continuously in the xylem vessel from the roots to all plant cells. 90% of water diffuses out through stomata. Transpiration also takes place through the lenticel. This means only 10% of transpirations diffuse through lenticel. 99% of water evaporates from the leaf and is lost to atmosphere. Only 1% of water is used by cell for photosynthesis and for turgidity. Now we look at the environmental factor that affects the rate of transpirations. There are 4 environmental factors: light intensity, temperature, relative air humidity, and air movements. This is the graph show how the light intensity affects the rate of transpiration. This one is temperature, relative air humidity, and last one is air movements. Light intensity. From the graph, we can see as the light intensity increase, the rate of transpiration also increase. But at a certain point or level, the rate of transpiration is constant. Why? Because the relative air humidity, temperature, and air movements become the limiting factor. Temperature. From the graph, we can see that when the temperature increases, the rate of transpiration also increases because the kinetic energy of water molecule increasing. Relative air humidity. From the graph, we can see that when the relative air humidity increases, the rate of transpiration decreases. The graph going down. Okay, because when the relative air humidity increases, water vapor escapes slower from the stomata. I give you example. 7 o'clock at morning and 12 o'clock at noon. Which one is high relative humidity? 7 o'clock at morning. So rate of transpiration is slower. But at 12 o'clock noon, the relative air humidity is low. So the rate of transpiration is higher. Air movements. From the graph, we can see that when the wind speed increase, the rate of transpiration also increase because the movements of air carry away water molecule or water vapor that has been diffused out from the leaf. This is the example of experiments that we can conduct to study the effect of environmental factor on the rate of transpiration. In this experiment, we're using a photometer. The problem statement is, what are the effects of environmental factor on the rate of transpirations? The aim of this experiment is to study the effect of environmental factor on the rate of transpiration using a photometer. Here are the hypotheses. A. Transpiration rate is higher in the presence of wind. B. Transpiration rate is higher at the higher light intensity. C. Transpiration rate is higher at the higher temperature. And D, the last one, transpiration rate is higher at a lower relative air humidity. Hypothesis is a general statement only, so we need to prove it whether the hypothesis can be accepted or not. So how to prove it? By do the experiments. So the variable is manipulated variable is environmental factor. So we need to study 4 different environmental factor in these experiments. The responding variable is rate of transpirations, how environmental factor affects the rate of transpiration. And last one is constant variable, type of plants. We need to fix what type of plant we use in this experiment so that we can see how the environmental factor affects the rate of transpiration. Material that we can use in this experiment: leafy twigs, water, petroleum jelly, plastic bag, and tissue paper. Apparatus: cork, beaker, stopwatch, ruler, electrical table fan, 100-watt bulbs, and photometer. This is the photometer, and this is how we set up the apparatus and the materials. Photometer is a device used to measure water intakes by a leafy twigs. Procedure: number 1, prepare a photometer and ensure that the connections are applied with petroleum jelly so that they are airtight. Number 2, cut a leafy twig of a plant in water. Okay. Ensure the size of the stem is compatible with the cork covering the photometer. Number 3, set the leafy twig to the photometer and make sure it is airtight. Number 4, wipe all the leaves until they are dry using tissue paper. Number 5, fill the photometer with water until it's full. Number 6, strap a small air bubble at the base of the photometer to be used as indicator. Number 7, marks the initial position of the air bubble in the photometer as P. Procedure number 8. Place the photometer in a room without fan circulation and follow by placing it in front of the fan with speed 2. Okay, this one is to study the effect of air movements. After 10 minutes, after 10 minutes, measure the distance of a bubble in the photometer using a ruler or a scale on the capillary tube of the photometer. After 10 minutes, we fix it to 10 minutes. And then repeat step 1 to 8, by exposing the photometer set under the following surrounding. A. High and low light intensity. We put the photometer in a dark room and under the 100-watt bulbs. B. High and low temperature. One in room with air conditioner and another one in a room without air conditioner. C. High and low relative air humidity. One plant that cover with a humid plastic, another one will not cover it. And then number 11, procedure number 11, calculate the rate of transpiration. We use this formula. This is the example of table that we can use to record the result. Surrounding factor, distance traveled by a bubble in 10 minutes. This one we get from the result of the experiments. Transpiration rate, this one we have to calculate using the formula. Air movements, light intensity, temperature, and relative air humidity. This is all the environmental factor that we study in this experiments. Here are the expected results. When air movement is fast, the distance traveled by a bubble is 3.7 centimeter. When air movement is low, 1.5 only. High light intensity, 3.3. When low light intensity, 1.1. High temperature, 4.1. Low temperature, 1.5. High relative air humidity, 0.7. Low relative air humidity, 2.4. So this is the transpiration rate. This one you need to count using the formula. Distance divided by minutes. Discussion question. Why does the leafy twig has to be cut in water? To prevent air from entering it that cause the release of bubble into the photometer. What is shown by the distance traveled by air bubbles in the capillary tube of the photometer? The release of water vapor represents the rate of transpiration. Question number 3. What is the effect of air movements, light intensity, temperature, and relative air humidity towards the distance traveled by a bubble in the capillary tube? Air movements, light intensity, temperature increase the distance traveled by a bubble, while high relative air humidity reduce the distance traveled by a bubble in the capillary tube. Question number 4. State the operational definition of transpiration. You must give definition based on the experiments. This one we call it operational definitions. So the answer is like this. A process of loss of water. Transpiration is the process of loss of water from leafy shoot, which is shown by the distance traveled by a bubble in 10 minutes, affected by light intensity, temperature, air movements, and relative air humidity. You cannot give general definition of transpiration. The conclusion. Are the hypotheses accepted? Yes. Based on the experiments, all the hypotheses can be accepted. Okay. Okay, formative practice 2.3. Here are the question. Question number 1. What is the meaning of transpiration? State the relationship between transpiration and stomata. Question number 2. Other than higher temperature and fast air movements, light intensity also affects the rate of transpiration. Explain. Question number 3. A plant X was submerged in flood for 2 days. When the water subsided, the leaves of the plant X were covered with mud. How does this condition affect the rate of transpiration? Explain. Okay, we discuss the answer together, okay? Okay, question number 1. What is the meaning of transpiration? State the relationship between transpiration and stomata. Okay, the answer is transpiration is a process of water loss in the form of water vapor through evaporation from the plants to the atmosphere. Transpiration occurs through stomata. Question number 2. Other than higher temperature and faster air movements, light intensity also affect the rate of transpirations. Explains. When the light intensity is high, the temperature of the surrounding also increases. This causes the water vapor in the air to evaporate, and there is more space to be filled up by the water vapor that evaporate from the leaf. Hence, the higher the light intensity, the higher the rate of transpiration. Question number 3. A plant X was submerged in flood for 2 days. When the water subsided, the leaves of plant X were covered with mud. How does this condition affect the rate of transpiration? Explains. Muds that stick on the lower leaf will close the stomatal pore, and this will reduce the rate of transpiration. That's all for today. See you next lesson for subtopic 2.4, main organ for photosynthesis. Thank you and bye-bye.